So we completed the basic concepts of cell cycle the earlier day, and we understood that cell cycle can be defined into G1, S, G2. So this gap 1 synthetic phase and gap 2 phase are together known as interphase. And then we have this mitotic stage. And we also understood the earlier day that there is this transient phase present in cell cycle, that is the G0, the gap 0 phase. We understood the relationship of the changes in chromosome and DNA content during the cell cycle. Now, as last day, as I started my class today with the fact that, so every cell cycle passes through G1, S, G2, and then N. So that is the life of a cell. But there are always exceptions to a rule. And thus, we also have certain exceptions regarding to this defined rule in case of embryonic cell division, in case of skin fibroblast, and in case of oocytes. When we are talking about embryonic cell division, early embryonic cell cycles rapidly divide the cytoplasm of the egg into smaller cells. And it has been seen, as you can see in the picture, the embryonic cell cycles rapidly divide the cytoplasm into smaller cells. And in the process, if you look into the cell cycle that is happening, the cell is moving through mitosis and S phase only. The cells lack G1 and G2. The cells lack G1 and G2 because, as I said, this cell cycles rapidly divide the cytoplasm of the egg into smaller cells. So the basic aim of the cell cycle in this case is to divide it as fast as possible. So we have this S phase where the DNA doubles and the M phase where the DNA is again divided into two daughter cells. Again in the daughter cells, the DNA doubles and these daughter cells again divided into two granddaughter cells. So this is how the cell cycle goes on in case of embryonic cell division. There is no gap 1 and gap 2 phase. Now coming to another category of exception that is seen in case of cell cycle is the presence of G0 arrest in skin fibroblast cells. When we are talking about skin fibroblast cells, we are talking about the skin cells of our body, skin of our body. When a child reaches maturity or reaches adulthood where the maximum cell surface can be achieved, the fibroblast cells of the skin stops dividing because they have achieved the maximum surface area. Now what happens? These cells are living, but they have stopped dividing. They are able to stop dividing because at that point, the cells do not receive, or there are no growth factors present surrounding the cell. If you remember last day's class, I had said that growth factors are the food for the cells. So they are very important. And the cells need to have these growth factors for the cell to move from one stage to another. So when the skin attains the maximum surface area and there are no more growth factors available in the microenvironment, the skin fibroblast cells stop dividing, stop moving through the cell cycle, withdraw themselves into the G0 quiescent state. It is a quiet state. Quietness is there. If you remember, last I gave you this example that whenever there is a cut wound, blood starts oozing out. With blood comes platelets. Platelets produce these platelet-derived growth factors. And the moment the growth factors are again being perceived in the environment, the skin cells surrounding that cut wound come out of the G0 state, start dividing, and provide a fresh skin over the wound. Once again, that fresh skin has been synthesized. What happens? No more growth factors again available, and these cells again move back to their G0 quiescent stage. And the final example of exception to this rule is the G2 arrest for oocytes. All females, when born, are born with a certain amount of oocytes which will mature in their lifetime to give rise to eggs. Now, as long as puberty is not attained, the oocytes remain arrested. With puberty, one after another, the oocytes come out of their G2 arrest, mature, and are released as egg. So these were the exceptions of rules to cell cycle. And as I said, a growing oocyte is arrested in the interphase. Progesterone signals the oocyte to pass through meiosis 1 and meiosis 2. It becomes arrested in meiosis 2, awaiting fertilization. So oocyte is arrested in meiosis 2, that is G2, awaiting fertilization. Once fertilization occurs, the second cell cycle, once fertilization occurs, the cell can come out from this second cell cycle arrest, and then the fertilized egg moves to proceed through the embryonic cell cycles. So that was the story of a growing oocyte that is arrested in interphase or G2 phase. And now we come to another point, that is the stages of cell cycle, and more importantly, we have now understood the stages of cell cycle, but more importantly, the molecular players of the different stages. But prior to that, let us look into this picture. This is a comparison between yeast and higher eukaryotic DNA. As you can see, in case of yeast also we have this G1, S, G2 and N. In case of higher eukaryotes as compared to yeast, as you can see, the G1 phase has increased. So we have G1, S, G2, M. And in both the cell cycle, you can see that there is a dark dash present through G1, which is a very important checkpoint. We will study in details what happens at the molecular level, what happens during this checkpoint. And this checkpoint is known as restriction point in case of higher eukaryotes and the start point in case of yeast. For a cell to pass through this restriction point or checkpoint, the cell has to ensure that there is no DNA damage, that nutrients or food is available. And since this picture diagram shows about yeast also, regarding yeast, the mating factors must also be present. Regarding higher eukaryotes, the cell checks whether there is any DNA damage or not, or whether nutrients are available or not. So assuming that there will be no DNA damage and all nutrients or food materials are present, the cell passes through the G1 to S via the restriction point. So once more, the restriction point is called start in yeast, and the checkpoint is called start in yeast and restriction point in early G1. Now, as I said, let us look into the molecular players of this cell cycle. In this picture, as you can see that in each stage, there are two partner proteins. One is cyclin dependent kinase, which is briefly written that as CDK4, and the other is cyclin D. We will be getting into the details of CDK and cyclin D, the cyclins are functioning at the molecular level further on. But for now, just remember that these are enzyme protein partners. Cyclin dependent kinase, you have already studied cell signaling, so you can understand that this is a kinase which phosphorylates its substrate. but the activity of this kinase is controlled by a partner regulatory protein, which is known as cyclin. Each stage of cell cycle has its own set of cyclin and cyclin dependent kinases. So we have CDK4 and 6 cyclin D partner, which helps the cell to tide over the G1 restriction point. At the end, on the entry of or prior to entering S, we have the cyclin E CDK2 partner. During the synthetic phase, we have cyclin A CDK2 partner. The transition from S to G2, cyclin A CDK1, and the transition from G2 to M and coming out of M, controlled by CDK1, cyclin B. So cyclin and CDK is the enzyme part, cyclin is the protein regulatory part. These two act as partners or pairs in different stages of cell cycle, bringing about the defined control of cell cycle as we see it. So once we are thorough with that, these are the partner proteins and the regulatory partner proteins and enzymes that are present through the different stages of the cell cycle. They are not only present, they exert an extreme molecular control over what is going in that particular stage. We now move on. We start with what is happening in late M and early G1. So we are now going to enter the stages of cell cycle. So if you look into this particular picture, late M, early G1. So this is that stage I am now talking about. Once more, so we are actually going to discuss what is happening here. And in this picture, as you can see, the point where I have circled, you're not seeing at that given area any cyclin dependent kinase or cyclin molecules controlling there. So this is a small window that is present during late M and early G1, where the function of cyclin and their corresponding enzymes, the cyclin dependent kinases, are very low. And in this window, something very, very important happens. So what happens is that it is in this window that the DNA is prepared for replication. Mind it, replication does not start unless and until the cell enters the S phase. But the window I just now talked about, it is during that window that the DNA is prepared for replication. Since you have done DNA replication in your semester 1, you remember that for replication to initiate in DNA, there are certain points which are recognized, and in these points, the DNA double helix opens up, and the replication machinery sits on the replication fork, and the replication proceeds in a bidirectional manner. This is something you have already studied. When we are talking about eukaryotic replication, so in a very similar manner, to initiate DNA replication, a pre-replication complex known as pre-RC. So a pre-replication complex must be assembled at points from where replication will start. So these points are known as origin of replication. So in this origin of replication, or the sequences of DNA which are recognized as origin of replication, they are recognized by a complex of 6 protein, which is known as the origin recognition complex. So the cell has just come out of M. The cyclin and the CDK partner of G1 has not yet taken control. A small window is present where the activity of G1 cyclin CDK is low. During that time, the origin of the DNA are recognized by this origin recognition complex. The moment the origin recognition complex binds with the origin, it then serves as a platform for binding of two more important proteins, as you can see, CDC6, CDT1. So next, after ORC has attached with the origin, now comes CDT1 and CDC6, which now binds with the origin. Once this has also been completed, so CDC6 and CDT1 binds with the origin. This usually occurs during late mitosis to early G1. This is important. So this is the loading of the origin recognition complex to the DNA. And as you can see, it has been mentioned here, this occurs in that small window. It is occurring here. Occurs during early G1, late mitosis stage. So once the origin recognition complex followed by CDC6, CDT1 has been loaded, next comes the mini chromosome maintenance complex, MCM, which is a ring made of 6 closely related proteins. And this is the eukaryotic helicase. So you can see MCM comes and binds to this complex. So this is now the pre-replication complex. And the pre-replication complex is now ready for activation. It is not activated, mind it. It is not activated, it is ready for activation. The moment the cell will cross the boundary of G1 and S, this structure will be activated. So this is one very, very vital thing that occurs in the small window of early G1 and late mitosis. As I again said, and I'm again repeating, that this pre-replication complex formation is restricted to the window of the end of M phase and the early G1. As you can see, so this is a form of a licensing system that licenses the DNA and tells when to start replication. What happens next is that the moment the pre-replication complex is formed in early G1, the complex sits in this manner and the cell passes through the G1, crosses the restriction point and is on the verge of entering the S phase. The moment it is on the verge of entering the S phase, you can see CDC6 has been attached to the pre-replication complex. And now you can, what you can now see is that for this structure to enter replication, inactivation of CDC6, CDT1 and ORC is done. So these three proteins are inactivated, they open up from the DNA and DNA is ready for replication. So it enters S phase. So in other words, CDT1 and CDC6, these two proteins are known as licensing proteins. They give license to the DNA and tells them when to replicate and when not to replicate. As long as they are bound to the origin with the ORC, they prevent DNA to initiate replication. They are part of the pre-replication complex. But the moment the cell crosses the restriction point, these three molecules are inactivated, biomolecules are inactivated. And in short, a license is now given to DNA that you start replication. So we understood the licensing of replication, and we also understood that the replication phenomena is very controlled. Unless and until the cell crosses the restriction point and is on the verge of entering the S phase, even though the DNA is ready for replication, the replication doesn't start. Question is, why doesn't it start? Because as long as CDC6 and CDT1 and ORC is bound to this pre-replication complex, replication cannot start. They only open up when the cell is on the verge of entering the S phase. So this is a beautiful system of ensuring that replication starts only at S phase. But how do we ensure that it stops at S phase and does not restart before G1? So once replication starts and replication proceeds through S phase, if you remember, MCM is a helicase. So it ensures it gradually opens up the DNA, once it has opened up the DNA, it leaves the DNA. And by that time, all the DNA has replicated. To start a fresh batch of replication, we need to have the MCM again. Because as I said, MCM is a helicase. And MCM cannot bind to DNA at any point, at any time and anywhere. For binding to DNA, it needs to have this complex of ORC, CDC6 and CDT1. And these three molecules can only bind to DNA at this time. So even the MCM is present, cell has existed the S phase, the MCM doesn't initiate replication because its loading is restricted to this phase only. So an important take-home message is that there is two paths to DNA replication. One is loading the DNA for replication and one is actual replication. Loading the DNA for replication is essential because unless this happens, replication will not start. Loading the DNA for replication is totally restricted to late M, early G1 window. DNA cannot be loaded at anywhere in any other part of the cell cycle. And the actual start of the replication is again restricted to the S phase. So even though the DNA has replicated in S phase, it cannot again initiate fresh replication because loading has to occur in the next cycle of late M and early G1. So this window is very important. So this is one. As I said, there are two phases of DNA replication. One is the G1 phase loading and one is the activation phase. Helicase loading allowed only in G1, but the activation of helicase is not allowed in G1. We discussed that. And again, loaded helicase activated in S phase. Once the activation has occurred, replication has occurred, the helicase has opened up. Fresh loading cannot take place, as you can see, in S, G2 and M phase. Fresh loading is not allowed in these three phases. So loading phase, as I said, a small window where the cyclines and the CDK dependent kinases were low. Helicase loading is occurring, and this picture beautifully summarizes. See, helicase loading is occurring, but no helicase activation. The moment the cell crosses the restriction point in S phase, helicase activation, no helicase loading, no helicase loading allowed anymore. See, it's passing through G2, it is passing through M. No helicase loading. Again, only when it reaches a phase of this window, then only helicase loading is permitted. That is permitted by CDC6 and CDT1 because they come and recognize and bind to this ORC and then they allow the loading of helicase. And hence, CDC6 and CDT1, as I stated earlier, is known as licensing proteins. So this is the tight control that is kept on replication during the stages when the cell passes through the different phases of cell cycle. So we understood the strict control, how the DNA replication is kept in check during the stages of cell cycle. This is, let me just say. This is another beautiful picture. This picture is from Bruce Albers. You can see Origin recognition complex, the one we discussed, it is a recapitulation of the same. The CDT1 and the MCM helicases, they're coming and binding. We are having the pre-replication complex, G1, S phase, entry of S phase. The entry of S phase is marked by the inactivation of these 3 proteins. And you can see start of replication. When the cell is moving out of S phase, DNA replication completed, but as I said, loading cannot be allowed. So unless and until helicase loading is allowed, the fresh cycle of replication cannot be initiated. So this is how the replication of DNA is controlled through cell cycle. And now in the next class, we have passed through that phase, late M, early G1, where we have understood how the control over DNA replication is maintained. Now the cell will move gradually from G1 restriction point to S. So this point is again very vital. We'll start this discussion in our next class.